Welcome to the data analysis training. In the previous video, we have talked about experiment design and ran the image analysis workflows. Now we have data tables that contain the median signal minus background values for each peptide, array, cycle, and exposure time. During data analysis, we further transform these values. In this video, I will talk about the first part of the data analysis, which is called basic processing. This includes first the quality control or QC of the peptides, which aims to exclude low-quality peptides. The second step is the normalization of the peptides that have passed the QC. Now, let's go to Tursin. I am working from the PamGene Data Analysis Training Team, Data Analysis Project. You guys, however, please work from your own team in the same project where you have done the image analysis. For this training, you also need to run the PTK afterwash workflow because the export file is also needed for the PTK data analysis workflow. Let's add a new template. We need to select the correct template type based on the experiment design. Now I will select the CT1, T2, T3 template. Let's take a quick overview of the beginning of this template. It starts with uploading and joining two data tables. One is the export from the image analysis, and one is a data table called enrichment. This is an annotation file for the samples. Next, there is a step called ETS, which is the exposure time scaling. It combines median signal minus background values at multiple exposure times to create a single value per cycle, which is the S100. Next, there are three steps that concern the quality control of the data: the mean and CV, the prephosphorylated peptide, and the PTK QC. These are the steps that are involved in the first part of basic processing, the quality control. Now, let's take a closer look. First, I will explain how the enrichment table for the samples should be made. You can see the two designs here. They contain the samples below one another. They always have the columns barcode and row, since these are the columns based on which the enrichment file is matching to the image analysis export file. Next, we have sample name, test condition, and replicate. As you see, one replicate with all its test conditions is on one chip. The last important column is called supergroup, which denotes the model system. In a simple PS12 project with 12 samples and 4 test conditions, there is one model. But in the experiment design on the right, you have two models, thus two supergroups. Let's see a more concrete example for this. In the first experiment design, we are working on HeLa cells, and as test condition, we have 3 drugs and 1 untreated PBS control. The sample name is unique, and it contains the test condition and the replicate number. Below, in the second experiment design, there are 2 cell lines, HeLa and HEK293, and only 1 test and a control condition. To make the sample name unique, we need to create it from the supergroup, the test condition, and the replicate number. Importantly, we set up our workflows to click as little as possible. Therefore, we need to follow some rules in the sample annotation, which you can see here on the right side. I have already told one that the sample name has to be unique. Then, you always need to have supergroup and test condition columns with the same exact names, for example S, group 1, control, T1. Be careful that you don't make typos. However, you also need to add your annotation as well, which you can do in separate columns. We name these columns as supergroup_annot for annotation, but you can name these columns differently. Here you can add the real names of your conditions. We suggest to give short names, since they will look much better on heat maps. And, of course, you can have additional columns if it comes handy for you, for example the date of the run or the experiment number. Next, let's upload the sample enrichment file to the project. Click on New Dataset, Delimited Text, and Upload. Importantly, you have to check if the columns have the correct type. They can be numeric or character. There are two reasons why they have to have the correct types. One is for joining two data tables. They can only be joined on a column if they have the same type. Since image analysis outputs barcode as character and row as numeric, we have to make sure that these match with the sample annotation file, since the image analysis output and the sample annotation is joined together with these columns. So I will change the barcode to character. The other reason why we have to set the correct data types per column is because the Y factors in the crosstab view have to be of numeric type. They cannot be characters. Therefore, it is also good to not have any additional numeric types, which should be characters. Let's upload this and go back to the workflow. First, let's add the data tables. For the PTK workflows, we have to select both pre-wash and after-wash together and upload it. Let's upload the enrichment file that we have just added. Here, in the join step, you can see that they are joined by barcode and row. The next is the exposure time scaling. You can see that this step looks a little different than the other steps. There are two rectangles, and on the top left corner there is a different sign. This indicates that this is an application. When we run it, a wizard pops up. The wizard asks to select which factors describe, for example, the quantitation type, which is the Y factor. Since ETS integrates the median signal minus background values from different exposure times, that is what we need to select. However, actually here everything is already pre-filled and we usually don't have to change anything, so we don't need to click through the wizard, but can just click the Run button. The output value of the ETS is the S100 signal. Next, let's talk about the pre-phosphorylated peptides. These are used as positive control to check the performance of the detection system, such as the antibodies. We don't always have to check this step. However, if in later steps we see that there is a sample with overall lower intensity, then it is good to check this. If pre-phosphorylated peptides have high signal, it means that there is enough antibody. Therefore, the lower intensity stems from biological variability. If the signal intensity of the pre-phosphorylated peptides is not good, this would indicate a technical problem that there is not enough antibody. In this slide, you can stop the video and check it out what other special peptides are on the chip. Now we arrive to the quality control steps. There are two of them, the mean and CV and the QC-PTK. Let's first take a look at the mean and CV step. The coefficient of variation, or CV, is an important quality metric. To run this, we have to have a somewhat special projection in the crosstab view. We want to calculate the CV per test condition, so we need to have multiple values for each test condition column, and we also have to color by this factor. If we have, for example, two PS12 runs with two model systems, then we also need to drag and drop the supergroup factor to the columns and to the colors. Let's run the app. On the right side, for each condition, we can see a CV versus mean plot, in which each is a peptide. There is also an error model fit to the data. The main output of this operator is below the figures. It is the CV high signals. This is the value of where the curve fit becomes flat. For biological replicates, if this value is under 30%, we consider it good data, meaning that the amount of biological variation is not too much. Comparing two conditions with not a lot of variation would probably yield a lot of significant data. Till 40%, the quality is fair, and above that it is poor. Later, I will show you a table where this information is summarized. On the left side, you can see that there are some parameters of the error model fitting that you can adjust. We suggest that you only adjust it when you have an NA for the CV high signals in one of the conditions. That means that the model could not be fit on the data, but adjusting these parameters within the given range is accepted and can solve this problem. However, when the model can't be fitted initially, the CV would be anyway very high. We suggest that in your reports, make a note of the quality metrics of your experiment and record these CV values. Next, let's take a look at the PTK QC app. The aim of this step is to flag those peptides which are not present in the samples, so in the next step, we can filter them out. Let's run it. Again, in the wizard everything is filled out correctly as default, so we don't need to click it through. However, there is a special case when we do need to change the default cycle number here in this filter. The PTK QC app uses the information of which cycles are considered pre-wash. If the experiment run has been stopped, then the cycle numbers would change. You have seen how to adjust filters in this case in the image analysis workflow. In the data analysis, you have to do the same in this step, to just replace 94 with the number of the last cycle in your data. Let's run the step. When the app has run, we can double-click on it and see that there are two visualizations. Let's click on the kinetics colored by presence. So how does the PTK QC app find which peptides are present or not? First, it calculates a presence value for each peptide S100 value per barcode and row. The presence value is positive if peptide signal shows positive trend in time, so throughout the cycles. The cycles are added as the x-axis below barcode and row. Here we can see what is the trend of the S100 values through time or cycles. Orange values mean positive presence, and you can see that those peptides that show a positive trend are orange, while no trend or downward trend is colored blue. Those peptides are not present. Importantly, there is a row factor here called fraction present. This is the fraction of peptides which are present, that is, the fraction of those samples that show a positive trend. Since this factor is the first row factor, the peptides are sorted based on this. You can see that more blue peptides, which show up in less conditions, are on the top, and the orange ones are at the bottom. Let's check out the other visualization of this app, which is the presence map. This is very similar to the previous one, but it is a heat map of presence values, so it is easier to have an overview of peptides with different fraction present values. In the following steps, we will use this fraction present value to exclude peptides which are present only in less than 25% of the samples. Importantly, this threshold of 25% is based on an experiment with 4 conditions, since 25% is one condition, and there is a chance that all the not present values belong to one condition. So this threshold allows us to keep only those peptides which are present in more than one condition. Therefore, we should always revise this threshold based on the experiment. For example, if there are more than 4 conditions, or if we would expect that there can be important peptides which show up in only one condition. For example, in an experiment with very potent inhibitors, the phosphorylation of some peptides could decrease to an extent that they would be only present in the control condition. But we would still be interested in them. For PTK, QC is done based on the signal kinetics throughout the cycles. However, for SDK, we do not have this information, so we need to rely on a different method. So we make use of the coefficient of variation again. Here is an example for an SDK QC app in an SDK workflow. I double-click on it to see the visualization, the log signals by nominal CV. The app calculates an estimate for the nominal CVs for each peptide across all samples. This is called the pep.cv estimate, which you can see as the first row factor here. Low CV estimates coincide with high signals, which are on the top. As we scroll down to higher CV estimates, the signal is lower. In the next steps, we need to exclude peptides based on a threshold on this pep.cv estimate factor. This threshold is 0.5, meaning that we exclude all peptides which have CV higher or equal to 0.5. Importantly, again, we always have to revise this. Most of the time, 0.5 is a stringent threshold, and we do not get sufficient number of peptides passing it. Therefore, please carefully inspect this heat map. If the values on the heat map a bit higher than 0.5 have similar values as the ones just below 0.5, you can set the threshold higher, making it less stringent. For example, in this case, I would set the threshold at around 0.8. Let's go back to the PTK workflow, and I will show you how excluding the peptides works. After the QC app, we can choose from two kinds of normalizations, which I will talk about in a bit. For now, let's look at the log normalization. In the filters, the QC filter is already added. Fraction present is higher than 0.249. You can already see that even though the log operator here is not run yet, the filter is already working. On the bottom of the page, you can see the number of rows, which would be 195 if nothing would be filtered out. In this slide, there is a summary of when the QC thresholds are needed to be adjusted. You can pause the video to read it. Finally, let's summarize the two different QC metrics we have talked about. First, using the mean and CV app, we assess the variation of the peptides. Second, using the QC app, we excluded peptides which were not present. Based on this, we can give flags to the data. The data quality can either be good, fair, or poor. You can see the thresholds for the number of peptides passing the PTK or SDK QC and the CV for technical or biological replicates. These quality flags serve to assess the reliability of the data. From the basic processing, we have discussed the quality control. The next step is normalizations. Here is an overview of the normalizations in a workflow. We can choose from two basic normalizations, log and VSN. The workflows include both of them. As we have seen before, the log normalization is together in one step with the filtering for QC peptides. However, if we do VSN, the filtering for QC peptides is in the next step because VSN takes all peptides into account. There is one more optional normalization to correct for batch effects. This can be done for upstream kinase analysis or for other multivariate analyses. Let's go one by one. First, I will explain the log operator. Then, I will talk about what is VSN and when it is better to choose that instead of log transformation. Let's look at the log step. The operator is called log cutoff because additional to the log transformation, it also handles the negative result values that come from values between 0 and 1. If we go to the Settings tab, we can see the parameters of this operator. The treatment of negative values is set to mask at cutoff, and the cutoff is set to 1. This means that the operator assigns the value of the cutoff, 1, to the input S100 values which are below 1. In this way, all S100 values below 1 will have log transformed result values of 0. Additionally, there is a third parameter, log base, which is 2 by default. Let's also take a look at the filters. Importantly, there is a filter coming from the PTK QC app, the fraction present higher than 0.249. You can also see that the number of rows is lower than the number of peptides on the chip. This is also because there is an additional filter to get rid of the positive control and the reference spot. Additionally, we also filter for cycle 94 since the previous cycles were only needed for the QC app. Now, let's go back to the template and run the step. When can be VSN useful? In log data, the variance is higher for features with low mean. Therefore, the same fold change for a peptide with low mean is less significant than for a peptide with high mean. Variance stabilizing normalization, on the other hand, leads to data where the variances are similar for features with low mean and high mean. The 4, VSN facilitates the discovery of significant peptides with low signals. However, we have to be careful when applying VSN since the interpretation of fold changes will change. This is because VSN contains a step in which the data is transformed in a way that results in a shift of the y-axis to the data mean. Therefore, VSN reveals the relative differences compared to the mean of all samples, not the absolute differences. Here is an example here how that works. Let's take a dataset for which a t-test after log reveals a general upregulation of phosphocytes in test versus control. If we do a VSN on this data, a subsequent t-test would result in a V-shaped volcano plot. Here, we can see which phosphocytes show lower or higher signals compared to the mean of all samples. Thus, the fold changes we see on this plot are not the absolute fold changes, but the relative fold changes compared to the mean of all samples. Additionally, VSN is equivalent to a log transformation, thus its output data is on log scale. Here is a text that explains how to choose between log and VSN. It explains when VSN can be a better choice. You can stop the video to read it. It is simple to run the VSN. We don't need to change anything. You can see that there are 195 rows in the dataset because VSN has to run on all peptides, excluding the positive control and the reference. The next step is to filter for QC peptides. Here, the filter for the QC threshold is already present. The Y factor is VSN norm, which stands for VSN normalized data. The operator is called identity. It just outputs the same values as in the input. However, when there is a filter in the step, it only returns those values that are in the step. In this case, the VSN normalized values will be only returned for these 178 peptides. Next, I will show how to make heat maps and PCAs from normalized data. This heat map app makes simple heat maps. We only have to set the quantitation type or Y factor in the wizard. Since the app is after the log step, we have to look for a factor here that would come from there. And this is S100QC, then a dot, then log transformed. I'll run the app, and while it is running, I will explain where the name of this factor comes from. Let's go back to the log step with a double click. In the environment, we can see the namespace, and this is S100QC. Thus, the output of this step will be S100QC, then a dot, and then something that is specific to the operator. Here, it is called log transformed. But how can we know what it is called? If we are unsure, the operator will tell us. Click on the version number below the operator. This takes us to the GitHub page of the operator, where we scroll down to the README, where we can read what the output relation is called. The output relations are the factors which are calculated in an operator. Here, it is called log transformed. In summary, here is how the name of the output of an operator is constructed. Namespace, then a dot, then the output name coming from the operator. The heat map is run. Let's double-click and check out first the simple heat map. You can see that the heat map is sorted from high to low log transformed values. The log transformed is in the y-axis. The sorting is done because the first row factor is the R order, that is row order. It is ordered by the mean of all values in a row. This is not needed in the figures, so I will decrease the size of this factor, as well as the ID. If we reset this step, we can drag and drop other factors on the crosstab view. For example, instead of barcode and row, we can add the sample name. Let's first delete these factors from the columns. You can see that there are now only 4 columns. In a heat map, These show the means of the 3 replicates. Now, let's add the sample name below test condition. So this is how you can customize the heat map. When you rescale it, you can see how the width and height changes in the bottom. You can click on Download to save it as an image. Let's check the other visualization in the heat map. Since we have reset one of the visualizations in the app, now it appears as if it is not run. Still, click on it and go to Scaled Heat Map. In this heat map, the z-score scaled values are shown between 1.5 and -1.5. These max and minimum values can be found in the colors and then palette. The Y factor is just called as scaled value. Let's see where this comes from. When we go just one step before the log heat map, we can see the app from inside. Apps are very similar to workflows. Indeed, they are sub-workflows. They always have an input step and a wizard, which is the pop-up we interact with when running the app. We can see that there are 2 branches, one for the simple heat map and one with an additional step called z-score scaling. Next, I will show how the PCA Shiny app works. You can use the principal component analysis, for example, to check how your samples are clustered. If your samples in a condition are similar, they cluster together. We can also use the PCA to check if there is a technical batch effect. First, we need to add those factors to the columns that we want to see in the PCA. Here, I will add the replicate instead of the row. Let's run it. There are multiple visualizations. I find the bplot as the most useful. It shows the variable loadings which we rarely use, so I decrease them. Now we can see that actually the samples do not cluster very well. Could this be because of batch effect? Let's change the factor for coloring to barcode. Now we can see that the blue chip clusters together, which is a clear batch effect. If you would like to document the data analysis, you can take a screenshot and save it. Additionally, you have to reset the PCA because it will keep running. Since there is a batch effect, we must take it into consideration in the downstream data analysis steps. Batch effects can cause high variation within a condition, so the statistical tests can be less powerful. For phosphosite analysis, we can do pairing, which counteracts this issue since we run the statistical tests within a chip. However, pairing is not possible for upstream kinase analysis. Therefore, we should counteract the batch effects. We can do this by Combat, a batch correction normalization. Combat, therefore, can be run before kinase analysis if the design is balanced. Let's see how it works. The input for the Combat Shiny operator needs a coloring factor, which should be the technical batch. It is barcode in this case. Let's run it. In the operator view, you can assess whether there are batch effects and whether they have been removed by Combat. There is a PCA done before the Combat on the right side and after the Combat on the left side. We can see that the PCA before Combat contains batches, while the PCA after Combat has removed them. So we can conclude that this has worked. So let's click on the Done button. After the messages in the right bottom have disappeared, it appears under the Done button, and then we can leave the page. Now the data that comes out of this step is Combat corrected. We can also check in the PCA how our samples cluster now. We can clearly see that conditions are better separated. You can compare the Combat PCA on the right side with the screenshot of the log PCA on the left side. In this video, I have introduced the upstream part of the PamGene data analysis process, the exposure time integration, the QC, and the 3 kinds of normalizations. These are very similar for all experiment designs. In the next video, I will talk about phosphosite and kinase analysis.